Hello everyone, I'm Suparna and today I'm here to discuss my work which deals with the prediction of binding affinity and understanding the mechanism of inhibition of FDA-approved compounds on hepatic transporters BSEP and MRP2. Now, hepatocytes are polarized cells with specialized transport systems on the canalicular and sinusoidal membrane, which maintain bile acid homeostasis and detoxify endogenous and xenobiotic toxins. Owing to these critical functions, interference of their functional transport processes can lead to intercellular accumulation of xenobiotics and bile components, which might result in cell damage as a result of cholestasis, which are often together termed as drug-induced liver injury or, in short, DILI. Of note, the bile salt exporter pump BSEP is considered the primary culprit for possible transporter-mediated delays, with MRP3, MRP4, and MRP2 proteins following closely on the list of transporters that are most likely to be involved in any kind of drug-induced cholestasis. In this work, we are mainly focusing our attention on the canalicular efflux transporters BSEP and MRP2 because clinical reports presently mention that around 30% of the clinical drug-induced liver injury cases have been primarily caused as a result of BSEP inhibition, which results in cholestasis with concomitant observations of conjugated hyperbilirubinemia as a result of MRP2 inhibition. While several experimentally validated compounds have been considered inhibitors for the BSEP protein, the lack of an experimentally validated structure for the MRP2 in the protein data bank limits the scope of extensive ligand binding studies with this particular protein. Consequently, in silico approaches in combination with homology modeling have unveiled the identity of candidate inhibitors which further favor a comparative analysis in between these two targets. Furthermore, computer-aided drug designing CAD approaches are important in reducing the experimental burden on in vivo testing on animals for designing of safer drugs and for repositioning of the already known drugs in the market. Briefly discuss the experimental strategy that we have adopted in our study. We have downloaded the structure of BSEP from Protein Data Bank and we have modeled the structure of MRP2 via homology modeling approach using the Swiss model. And we have obtained the list of inhibitors from PubChem, which were around 100 in number. This was followed by target and ligand preparation, and then we have docked the lead compounds onto the target proteins BSEP and MRP2. And further, we have then performed the post-docking analysis using Discovery Studio, and we have additionally also performed toxicity predictions using Vega. Finally, coming to the results section, uh, among the 100 compounds screened in this study, the top 14 compounds with most negative binding energies among the 100 were listed and ranked accordingly. Six compounds were found to be common for both BSEP and MRP2, and these were staurosporin, zafirleucus, clofazimine, itraconazole, tolvaptan, and glycidine. Among these six compounds, staurosporin appears to have the highest negative binding affinity for the two proteins BSEP and MRP2. Toxicity predictions that were performed additionally show that staurosporin is both mutagenic and hepatotoxic in nature, but it does not have carcinogenic properties. That is, it is non-carcinogenic in nature. Post-docking analysis of the target and the lead compounds have revealed that the dominating interactions between the target and the lead were pi-pi interactions and conventional edge bonding interactions. Also, staurosporin, like staurosporin, the other five compounds were docked onto the protein, and post-docking analysis was performed for all these compounds with the target leads. Now, finally, coming to the conclusion that we have drawn from our study. Uh, Among the 100 compounds that were shown here, it was a general observation that the compounds which have most negative IC50, most lower IC50 values from literature study were found to have most negative binding energies. The dog compounds had prevalent interactions of non-bonding and hydrogen bonding interactions, which further dictate the best fit the compound onto the protein's active site. Such in silico studies are therefore helpful in bringing down the number of drugs that are to be screened in an in vitro setup, thereby fastening the entire drug discovery process and cutting down the overall cost as well as manual human efforts. Studying hepatic transporters is extremely crucial because liver, as we all know, is central to all kinds of drug metabolism and distribution, and any kind of perturbation with transporters or liver enzymes can lead to serious consequences within the system. It is therefore important that the in silico studies, the results obtained from our studies, which are preliminary in nature, are validated down the line with minimum but in vivo studies need to be performed with these in order to say that our results stand true in an actual biological system. So, with that, as I'm about to conclude my talk here, I would like to thank my mentor, Dr. Ashwani Sharma, for providing me with this beautiful opportunity to present my work at the third Women in Bioinformatics and Data Science LA conference. I'm extremely grateful and thank you all for your patient hearing. Thank you.